Cool. So, hi everyone. Thanks for joining today. And today, I'm really excited to hear from Siyu, who I used to be lab mates with, and now she's a postdoc at Stanford with James Zou and Stephen Quake. And her research works is at the intersection of like machine learning, computational biology, cancer biology, and she's focusing on developing multimodal models for single cell and spatial data, such as the one she's going to present today called Coral. And previously, before this, she earned her PhD at Columbia as part of Kam Leong's lab, as well as Elham Azizi, where I am also being advised by. So I'll pass it off to you to start. And again, like feel free to leave questions in the chat or unmute yourself if you have a question, and we can kind of treat this more as a discussion rather than this one-way presentation. Right. And with that, feel free to go ahead.
Okay, yeah, thank you so much, Justin. Thank you so much for the kind introduction and also the invitations for this seminar. And I'm very excited to be the speakers and excited to share some of our recent project on the spatial multiomics data integration. And we code it as Coral. I think one of the very exciting part of this project is that we are able to do a multi-scale integration, and we can do a comprehensive analysis with the spatial data. So I will probably start from maybe remind of you that in the last couple of decades that single cell sequencing have the power to enable the unbiased categorization of the cellular heterogeneity, and we're able to dissociating the tissues and into the individual cells and using these technologies to profile with like whole genome level of the gene expression. And as shown here as like a very big matrix, we're able to use machine learning and many computational methods to do like dimension reductions and other sort of like methods to understanding like the similarity and the variance of the cells. But this probably like not enough. Even they show a lot of like interesting biologies. I think researchers are want to know what exactly the cells are communicating with their neighbors, because usually the cells are not isolated. They are having their locations and affected by their neighbor. And as this like paper have discussed, there's like four of like major factors that's related to these sort of spatial related points. And one, I think the cell compositions, and along with the how the cell are distributed and across this space, and which will forming the spatial patterns. And this related to about like the molecular patterns associating with this pattern, and as well as like the cellular interactions that consider as like a bag of the moleculars. And four of these factors results in mostly the functionality of the spatial regions. And in a lot of like analysis, like we're interested to know, like for example, the STEM niches or the cancer niches, which may be related to how the cancers are associating with immunocells, and or how immunocells are infiltrating into the cancer regions. So overall, there is like a lot of questions that related to spatial biology. And very fortunately, we have so many emerging and excellent tools and technologies that came out recent years, which can actually having the profiles like across multiple modalities, for example, from transcriptomics to the epigenomics, metabolomics, and proteomics. And in individual of the modalities, there's also like a variable technologies and platforms. For example, for the transcriptomics, we have firstly like a VISEN, which is a spot level technology, and later on we have VISEN HD, which increases their resolutions in the spatial level. But I guess the number of the genes that profiled are probably a little bit lower, and which is very similar to the morphage that have the subcellular levels, but the number of the genes probably just maybe up to 2,000 or like a little bit higher. And there's another case of like technology like DBIT-seq that can do composition of like chromatin accessibilities as well as like histone modification, and mass cytometries that can do spatial metabolomics as well as like codecs for the protein profilings. So actually, I guess like because of there more and more data came out, there's like a need that we actually want to integrating how to these data sets. And I think one of the major challenges in here is like how can we actually integrating them with different scales and also their different like profiling depths. And also because we want to actually study these modalities on the same slices, but the truth is like we could not actually having this modality like just on one. So we have issues with how to integrate in different slices, but maybe recent adjacent. And there have a lot of like great methods that came out in this recent months. For example, we have like like two of the representative methods like MISO came out in early this year and Spatial Blue in later last year. They're all very nice they're all very nice models. But I think their limitation for now is that they are only able to do the detecting of the spatial domains, and they requires that the samples need to be on the same resolution. So that will actually have some limitations. And especially, I want to maybe convince you that if you want to study the tumor microenvironment, which is a very complex scenario, we have like so many cells that are interacting with each others, and we probably more interested in like individual cells that very small for example, like the T regulatory cells that involve in this tumor microenvironment, we want to know where they are. So the domain probably like not the major thing that we want to focus in on. And also we want to know the gradient that actually happen in this space. For example, how the certain cell types that they are distributed along with the distances. For example, we from the tumor course to these invasive margins and then to the stromal regions. And this also may be related to a more complicated scenarios like how the cancer will initially develop and even have the metastasis stages. So all of them requires not just like the domain detection, but also need to really consider like how the cell individually working in these sort of like spatial patterns. And this sort of motivation are related to dissecting this microenvironment. And in our own cases that we are interested in like immunotherapy that happens in this microenvironment. So we want to actually using some sort of like method that can capture not the domains, but can capture the cellular related activities, and especially how the cells are communicating them with each other. A very nice example, I guess, also very traditional or like a very famous method that we actually using now to decoupling the sort of like complexity of the samples are using deep generative model. And these are very nice model because it's able to have a lot of like assumptions on the cells, and we know in what some sort of like marker latent variables can be modeled directly from these type of models. So the project of Coral is actually motivated by our previous projects. It's called Starfish, and this is also my PhD work that collaborating with two of very amazing lab mates and also our brilliant advisors. And this is related to integrating with transcriptomic data and the histology, and we're using that integration to decompose these cell types in the spot level of the vision data. So this is very powerful, but I would say like we are only able to get in the cell type proportions, and we also using the graphical probabilistic graphical model as well. So we want to actually having a extending version of the Starfish, and in this extended version, we want to not just like having the cell type proportion, but we can also study how the individual cells that are interacting with each other, and we want to utilize both of the advances in the modalities. So in this case, we trying to integrating the modality which having, for example, in modality 1, which having mostly the spot level resolutions, for example, VSEMs, or even the spatial ataxia that's probably very close to single cell, but still not exactly single cell. And we have a second modality, which are cell level resolution and, for example, codecs and cosmics. So if you're integrating like different modality and which has in different spatial resolution, so I think we need to consider some of the way in the model. But I think the four of the major like rationales is that we want to first utilize the advances of two modalities. Usually, for example, VISN, which so they have like very low resolution, they have higher number of the genes profiled, and for the codecs, for example, the spatial proteomics, they are not able to do like as many as the genes the profiled as the VISNs. They probably only limited in 50 proteins only. But they have like higher resolution, so we're able to do identify the single cell the types. So these sort of models able to come incorporating these sort of advances, and also at the same time we want to infer this cell activity individually. And I want to convince you that we the model itself, the four of the goal that we have are again integrating and inferring the cell-cell interaction, identify spatial organization, and deconvolving into single cell version, and as well as like identify the cell states. So now I want probably go a bit deeper about like how the model look like. And in this case, we having four of the observed data, and one is and this one is the single cell version data, we call it YI, and we have a spot level, we call it XJ, and we also have cell type that like identified from this YI, and we also creating a graph based things we call GI, and we also have like three latent variables. One is the spatial latent variable ZI that capture the spatial variation, and we have a one dimension spatial variable that's related to the ZI, but you can probably have more hierarchical features. And we have inferred in single cell transcriptomics XI. And if we're looking on the genetic models, it's related to traditional way that we have assumptions for these variables, and they are related to negative binomial. And for the single cell modality, we actually assume they are gamma distribution. And for the variables, we have the Gaussian as well. So we have two of the tricks like in the inferences model. One is the graphical attention networks. That is, we're trying to understanding how the spatial neighbors, they are able to interact with each other. And we have cross attention that try to capture the modality differences. Okay, so the overall of the models will maybe showing as this. Then we have two of the important. One is the spot level data modality, and the cell level modality. We integrating them and creating the graph. And we have the graph attention layer, and we have the cross attention. We have like a overall spatial variable, and in here the optimization processes. We actually not only having the reconstruction loss, we having the KL divergence, and we have two of the regularization terms that are trying to capture the heterogeneity, but also capture the spatial smoothness of the sample. And probably want to showing how the results look like now. And first we apply onto simulation data. So we compare with the simulation data that provided by spatial group first. And in here, they providing two of the modalities have the pattern. And so spatial group are able to have a very nice pattern recognitions compared with the ground truth. And if you're looking on their total VI, PZ concatenation, and MISO, and we've seen like they are not as perfect as enough. So Coral dealing something with different imports. We will downsample the modality 1. We will having a lower version, but like integrating modality 2 and downsample modality 1, we are having able to getting a very similar and comparable results with the spatial group. So this also showing from here as the scores, and much clearer that indicating their accuracies. So besides the domain detections, Coro can also do the deconvolving of the single cell transcriptomics, and we will directly just like comparing with our ground truth, and we will see like a very high value of the correlations, and we compare with the distributions, and as well we will see a very high resolution or high accuracies from the result.
Hey,
So
Siyu, can
besides
I
the
ask a question about the simulated experiment? So yeah, I
yeah.
really like these situations where you present kind of situation with ground truth where you can actually test whether it's working or not. I was wondering, could you describe a little bit more in detail what exactly the setup is here? Like the two modalities, are they intended to be representing this disjoint information about the ground truth, or is one just meant to be like noisier than the other? And when it comes to deconvolution, do you assume that you have annotations available for one of the modalities or both modalities? How do you actually get the cell type annotations aligned for the
Mhm.
deconvolution step?
Yeah, that's a very great question. So the data set is actually provide a spatial group. So I think based on their how they generating this simulation data, I think modality one is resemble mostly like a DNA, a gene expression distribution,
Mm,
and modality two are related to protein, because they are also trying to mostly like integrating the gene expression protein. So in my case is because when they doing the sampling, they will just like sample from like some specific patterns, not, I mean, for the protein, they using, I guess, Gaussian instead, sorry, gamma instead of negative binomial.
right.
And in our case, we would just downsample it, like using, like we will we will just like having average of like small patches as here, and then we having just average results on all the dimensions. So
Mm,
this is give us like the patterns here, and for the deconvolution, because we initially we have the design for individual cells like for here, so we will have a results, and we will making the downsampled modality one that will be back to a full version of the modality one. So because we have the ground truth, so we can directly compare in here, and yeah, so we will showing a high accuracy.
so would you say that Coral, because you're doing the downsampling, is effectively um, you're kind of comparing a more difficult task compared to the benchmark
Mm-hmm.
methods? Or do you think the down sampling actually has a little bit of a like helpful effect that it smooths over
Yeah,
the patterns? Because this data set
yeah.
seems like pretty structured.
Yeah, I would say like probably here from you can actually see the downsample already showing a little bit like good patterns. I guess like pattern detection is one of the tasks of the Coro. We actually indicate we also showing some results on directly integrating modality one and two, which are very similar as well. Probably I need to showing the results as well. We don't necessarily to working rely on the downsampled modality one. And but I guess like this part is some sort of unique features of Coro, because these methods like not able to deal with that. Yeah, so
Right.
I want to maybe convincing that because we can do multitasks, and here we want just like to resemble the scenarios that we met with, because usually we have a lot of like vision data, and we have some sort of like cortex data. Even we have like a lot of vision HD now. I think people, or like we have some data already that on the visions, and so we can able to analyze it. And I guess there's a lot of
Hmm.
space for us to, yeah, to explore in this the already existing data set.
Yeah, yeah, makes sense.
Yeah.
And maybe you'll get to this, but I was
Mm-hmm.
wondering if the model is restricted to kind of you have the assumptions of gamma and negative binomial, which kind of mirrors protein and then gene measurements, but
Yeah.
can you also do like kind of different mixtures? Because you were mentioning
Mm-hmm.
other um, versions of like low plex modalities, such
Mm-hmm.
as like cosmics, which is still
Yeah.
RNA but low plex. So in that situation, do you still use gamma, or do you
Mm-hmm.
revert to like a negative binomial choice because it's doing RNA again?
Yeah, that's a very good question. So I do have some other variants, like for example, dealing with ataxic, we having the options for the Poisson distribution. But I right now
Mm,
I'm not really having too much like kind of like choices because just to focusing on like the tasks we are dealing with. But I guess definitely for extended version or like for specific data sets, we will definitely providing that sort of distribution as well. Yeah. But
Makes
we're
sense.
showing the results on the person later in the
Okay,
slides. Yeah, cool.
perfect. Thank you.
Yeah. And yeah, thank you for your question. So for that is like the first like cases of the simulation data, and we also translate into another one on mouse cortex. And in this case, we have some just like tricks to focusing on merfish itself. And we have two of the version of the merfish. We have downsampled merfish and the truncated merfish. So for downsampled, we actually try different scales. And as you see, like we have in maybe have like a higher resolution one and like gradually go into a more coarse one. So I wanted just like showing how the coral can infer these spatial niches like across different scales. Actually also answer Justin's questions like before, because we will seeing definitely the scores will be changed, but still if you compare with the spatial glue and also other methods like Covet just for the domain detections that they are actually have much more higher on scores. So yeah, and the second
Siyu?
task that we sorry.
Yeah. So just like how do we infer the niches? It's like clustering on the latent space, the spatial niches.
Yeah, so it's the sorry, I probably need to go back a bit, but yeah, it's the space, it's the z variable here.
ZI. Okay.
Yeah,
And just like
yeah.
because we are like here on the graphical model, what do you
Mhm.
mean by VI, the 1D spatial latent? Like how do
Yeah,
you interpret
so
it?
VI is probably like a summarized version of the ZI because I want seeing like VI is like too complex and want to seeing like maybe a clean version of VI and indicating maybe like very interesting gradient related stuff. I will showing probably very soon about like how VI can actually help. Yeah. Um.
Super cool.
Yeah. So first up, I'll probably show how compare with a spatial scope. That is also another one for a single cell deconvolution, and we were seeing that the scale, I mean, coral with different downside scales, we are actually can be much higher than this spatial scope, and this also indicating from we compare with the synthetic modality 2, which have like lower numbers, so there you might probably not able to distinguish their clusters enough. But with the inferred high resolution modality 1 are having more separations between the groups, and we also confirm that with the cluster separation ratio. And then about in terms of the inferred V, I guess like they have something interesting about like gradient of or like the tendency or like a trajectory related stuff. So in these cases, we will see that the cluster is actually indicating a a tendency like from a a neurons cell types like from neurons to oligodendrocytes and along with the markers that are associating shifting. And so in this case, we probably not going that deeper, but we will show you some other utilize about how the infer V can give us in the biology interpretation. So besides these two of the major tasks, we also want to show you that we can do cell-cell interaction, and this is captured by the coefficient of the graph attention networks, and this is indicating how the whole of this data set look like, and the edges are mostly related to integration, and the nodes are the cells. We can summarize the whole maps here as these figures, and focusing on only the cell type itself, and we knowing what exactly the interaction look like across different cell types. And I want to go
For
in.
the interaction score, so is it
Mhm.
exactly just the attention weight in the graph attention transformer? Is that what it is?
Yeah, we will getting the average attention scores like between different nodes, and which means the cells.
I see. And is this a
Mhm.
directed score that you get back, or is it undirected?
It's probably like normalized, but yeah, it's just like the score values for now. We want to just like assuming the values here probably related. Uh, what happened? Sorry, is it okay? Yeah,
Yeah, that's good.
we want to show. Yeah, we want to show we want to show that the overall like the net interactions like between the cell types here.
Okay.
Yeah. So we want to show you another also still the simulation cases, and we're using assay design provided by our one of collaborator, and we have the ground truth of the cell types. So for and we sample from different cell types here, and we will just like having a version of like you know the sampled RNA expression, and then also have the protein expression. With that integration, the coral able to identify the niches which are very similar to the ground truth of our data, and this also indicating that from the UMAP of the protein which has low plex, and we will having a higher version of having an imputated single cell RNA, and that UMAP will give us like more separated clusters, as you clearly see that how they are performed. Okay, so now I want to maybe go a bit deeper about like how the corals can help us to discover some maybe biological related stuff, and we first want to focusing on the mouse thymus, and this also data sets provided by Spatial Group, and we want to compare whether we can find something more interesting than what Spatial Group has mentioned. So for about the thymus structure, there's a few of like very interesting components. Like for example, we have like medulla, which is mostly related to immature T cell zones for this development of T cells, and then they will come to the cortex with outer regions, so the cells will mature in these regions. And so we have two of the modalities. One is the RNA, and two is the protein. As we have expected that protein has definitely a more noisy version, and what we trying to do is like we first like down sample this one, so we will having a version like this. So and we also have a manual annotations, and this will based on a merging the spatial group clusters. So we have like a clean version of how these clusters related to specific biological regions. So we actually do multiple stuff. So we consider not downsampled it, but we also consider downsampled it. If you compare the results on the corals, we actually seeing they are very similar, and then they also indicating very nice like pattern related stuff or structures, indicating even like more fine structure that happens between the medulla and the outer region. If you're looking on the total VI and meso, they are probably like not that satisfied, and we having showing like higher scores compared with these methods, as well as like the more ones are that indicating more like spatial coloration stuff. Okay, so
in
so
this case, is a downsampling more of a test for coral? Because you have the high resolution information available, right? So in this case, are you mostly just downsampling to test whether coral can accurately impute the single cell data, or do you think it actually benefits in some way? Because I noticed the Moran's eye, assuming higher is better, seems like the low res actually achieves a higher Moran's eye score. So if you have any comments on that.
Yeah, I would say that's true. So I think the model is trying to claim that, I mean, we can dealing with like multi-scale. So whatever the scale look like, I mean, we can dealing with like the same the modality with the same resolutions, but we can also dealing with like with different resolution. So downscale is probably a easier way, because we can also kind of like compare with their deconvolution. But this is a case just like have the same resolution, but in most of the case we don't. So so in this case we having actually simulation part. Yeah, but for the future data sets, we also indicating how it can do for the real scenario case. Yeah, but this one are also related to we compare benchmarking different data sets. So we have the ground truth. Yeah.
Right. So what is the Moran's eye computing exactly? Like the Moran's eye metric, what is it computing?
Yeah.
What are you
so
testing
this
for?
this is scores for a lot of like spatial data, and it's like showing how the how these clusters can be mostly smooth cluster on the space, not like scattered. Yeah. So this is the major.
It actually does make sense that if you downsample it can actually have the effect of smoothing it out. Do you think that's why it's a higher score?
Yeah, I think so. Yeah. Yeah, we don't compare with a spatial group because the ground truth is actually a spatial group. So
Oh, I see. Yeah, right.
yeah.
Yeah,
Mhm.
got it. Thank you.
Yeah, of course. So then we want to also indicating how other variables that can give us. So for example, we have like inferred variable v, and there's something very interesting came out. Like for example, if you're looking from here, this is like your map of the protein, which is like highly mixed because the protein are very noisy, and we have the decomposed results. Here we're focusing on the downsample modality, and we will see like we're able to do much clean version of like the cell types that clustered by themselves. And if you're looking on the infer V, we're actually seeing a trinity that from the codex that from cortex, and then two of the separate directions. One is the connective tissue and another is like medulla. This is related to from the cortex to medulla or outside like to the connective tissue. Well, the usage of these sort of like figures, I guess, probably we are able to identify, from the gene expression that they are able to, seeing. Yeah, we are able to seeing that the change of like from the markers that related to cortex and then gradually to the medulla and connective tissue. And this is also confirmed with the region distribution change as well. So one of the interesting, because as mentioned that the medulla is related to the immature cell, so we are able actually to see that the key markers related to the cell maturation are actually showing a very nice changes, like higher in cortex, which indicating a mature stages of T cells, and then lower in medulla. And in the centers, we are also able to see the distribution of the dendritic cells that is existed in the mostly cortex region, in the middle region between cortex and connective tissue. And we also seeing another specific cell types that only existed in the medulla, and that is as you see that highlighted in these regions. And interactions are also indicating that there is some sort of things happen because the resolution here is pretty low. We don't have the annotation about the cell types, so we're not like going that deeper, but we want to study more on the tumor examples. Okay, so
can I ask a question about the inferred V?
Mhm.
So with these plots, are you taking just the actual cells, whatever their inferred V is, and then ordering based on that? I'm wondering how you generate these mean scaled expression plots on the right side, given that since the V is not cell type specific, I would imagine you reach zones that are extremely heterogeneous, and then I thought the ranges of values would be even more high variance than they actually are here.
Yeah. I really like the idea. So I think because probably like one reason because data here is like I think the change like from the cortex to the medulla is like more clean version, and also the T cell maturation is like the major functions that related to this issue. So it's probably like more clear. And the V here is like identify the most important like spatial variable things. So we will actually, I mean, even I just directly, just like range it as like a exactly values. I don't do anything, but like we still will showing that a very significant differences. For example, because cortex here like the V related to lower is like mostly cortex, so they have very high markers related to T cell maturation, and then in these medullary regions, so they are like lower. So I guess it will, in this case, we're showing that. But I would say like in like tumors, probably like the V here probably have like more complex complex functions because we will have to see more. Yeah, but in some cases, in the more simple scenarios, I would suggest to use, of course, to figure out something more more significant. Yeah.
Mm.
Yeah. Right. So we now trying to do something like in the real cases, and for example, this is like a hippocampus spatial metabolic mix, and it has like high resolution, but we also have a vision that compare like paired with this data. So the issue is that we don't have other information. So what can we do actually? And Coral able to integrating it. So in other data, in other like papers, sorry, for example, in the meso, they have to do using like external tools, for example, the eye stars, and then integrating with the histology and getting a high version of the hippocampus spatial transcriptomics, and then integrating again. So I would say like Coral just like can do it in the from end to end, and we are able to get kind of like deconvolution directly, not just like with any other external tools. So we are able to first like identify these patterns, and then we are able to see like a change like from in the inferred V that is correlated actually from like the CA regions to this DG regions in this mouse hippocampus, and we actually identify some key marker as well. Yeah, but we don't do a lot of like biological explorations on this sample, and we focusing on another one, this metabolic breast cancer. And this is the data also from the starfish. And from starfish, we actually define some of the hubs that related to this sample. We identify there's some sort of like hypoxia regions, and we have like a hub regions 3 and 4. We think there's like a T cell related and enriched area, which we will think these are hallmarks of the metabolic breast cancer. So Codex what it can tell us is like a very major cell types, because they're very limited on the number of the proteins profiled, and in most of the case, the immunocell they have subtypes, and we requires like more markers to be identified as the label to see those like subtypes. So in the starfish, we just trying to roughly to validating our proportion predictions, and we not doing like that further. So Coral are able to like trying to address these gaps, and I would say like the Coral are able to enhance the cell types that existed in the cortex, and we will actually identify like more fine-grained cell types, and we are also able to study the cell-cell interaction now, because previously we are also just have very rough prediction or like hypotheses about like how the cells are interacting with the major cell types existed in the regions, but now we are knowing what exactly they are. And the major cell types we are interested in the Treg cells, and we are very excited to see that actually the Tregs are interacting with the tumor-associated fibroblasts. And I think this is one of the hypotheses that happen that claimed by starfish, and we actually verify that from the coral. Yeah, so this is the one of the major findings that from this data set. And then we go into another type of data.
Quick question from the chat about the cell-cell interaction that you mentioned. Is there any incorporation of like ligand receptor databases in the score computation, or is it simply just the attention?
Yeah, so this value is actually just like the variables that inferred by the model, but for the inferred version, because we have inferred like single cell data, so we are also able to do cell phone DB or like other type of like methods directly and to identify the individual like molecular interactions. So we will see that later as well. Yeah, but for this is like a average or like a summarized version of how the models can inferred. And yeah, and I think like we want to just highlighting that we see mostly integration that Tregs with the tumor. Yeah. Yeah, and then we want to showing something that probably the first try on how we deconvolve the spatial ATAC data. So because I think currently we don't have this single cell version of the spatial ATAC, and can we actually using the Coral to achieving a single cell version of spatial ATAC? So this analysis like collaborating with Jin Tian from ARC Institute, and very excited to show that we first like focusing on one sample, which is from the DBIT-seq, and this label are annotated by integrating with the single cell ATAC-seq. Well, we say like it looks pretty good to show in the structures, but still very noisy. and we want to find to integrating it, and because we don't have any references, we first like finding references from the motherfish, very similar structures with these slides, and we trying to integrate with coral. So we using a method that developed by Jintian, which called like OQUALS. They are able to integrating our inferred spatial single cell ataxic with the references single cell ataxic. And very excitingly, we're seeing the nice merging between these two of the data sets, as these figures as the UMAP showing here. And then we want to when we want plotting it onto the space, we are actually seeing a very nice like regions related patterns that happens in our single cell version of the ataxic data. So for example, we are actually, this is major cell types identified, and this is like fine grained cell types identified. And the label here are annotated by integrating with the single cell ataxic. So we'll say like the inferred single cell spatial ataxic are biological relevant because they are nice to indicating the cortex regions and also the GABAergic like enriched regions. We also compare how our inferred spatial ataxic cell distribution are compared with the motherfish. We will actually seeing comparable between individual cell types. Not all of them are exactly the same, but most of them are. Okay, I see another chart. Okay, yeah, of course I will discuss that later. And beside that, we also have the comparison between the differential peaks using the SNAP ataxic tool, and we were seeing that we mostly coral inferred data can able to have comparable results with the single cell ataxic. So which means that we are able probably like to achieving a single cell version of the ataxic. Yeah.
So when you say integrating with coral, like
mm-hmm.
these are two data slices that actually weren't from the same sample. Is that correct?
Yeah. So the major
wow.
so the major thing is we first like getting. I mean, this is the major to confirm that the convolution of the coral is able to achieve like biological meaningful single cell ataxic. And I would say like this model is like very powerful because with the Poisson distribution, like here we usually to modeling this single cell ataxic, we're able to actually recap or deconvolve into like 150K peaks in here. And then we integrating with the real single cell data. Real single cell ataxic data.
Did you have any challenges with doing alignment of these tissues, or like did you have to change how many neighbors you would incorporate for a single spot or something? Because I imagine it's a pretty different case from the ones where that they come from adjacent slices or something.
Yeah, agree. So I definitely try different numbers of the neighbors. Actually, I try from like 5 to 30. I would say like the results maybe change a little bit, but they're actually stable. So yeah, quite stable.
It's cool.
But but it's running very slow because it's very huge data and we have like a lot of cells here. Yeah.
How long would you say it takes to train a single model of coral on something like this?
How long?
Yeah.
Yeah. So for this data, for this one, I would say it's pretty long. I think it probably takes a few hours. Using like one slide of like 100 H100. Yeah, but for others, we probably just take like less than 1 hour. Yeah.
Okay.
Ataxic is much more difficult. Yeah. Okay. So then, I don't know how much time I need. I left.
Yeah, there's like 15 minutes, but maybe we can save a little bit of time to discuss further about like the model, because I think especially the audience is more interested in the computational aspect of it.
I see. Okay, so I probably just maybe very quickly finishing in maybe 5 minutes.
Sounds good.
Okay, yeah. So okay, so I think like the these examples are actually indicating the power of like the models already, but we want to go much deeper about like the biology stuff. And as I said, like initially we are interested in how the microenvironment responds to the immunotherapy, and we are interested in like how can we actually dealing with the liver cancers, and as you know, like the lot percentage of the patients are not respond to the immunotherapy. So we want to know what exactly happened in this space. And we actually having data from our collaborator at MDA Cancer Center. And we have some of the cortex that compared with the VISM. So for example, here we're showing the cortex data, and they labeled by their major cell types. And then Coral able to identify this region, and with very detailed region domain detections, domain things like including the immuno domains and stromal domain, as well as like tumor domains. I highlighted some of them just based on they are more specific, and I will try to see like what difference they are. So here is the how the individual domains that associating with the proportion of the cell types, and we actually identified two of them are very interesting. Like one is the macrophage, another is like CD4 T cells, because they are corresponding to the non-responders in the sample. We first like seeing how they look like on the distances, the tumors, and in front here we actually identify interesting things that macrophages in the non-responders are actually more closer than the tumors, and which are very different with the CD8 T cells. And because of the deconvolutions, we are also able to identify many interesting, genes, because codex give us limited informations, and especially for those like very detailed expressions, and they probably like in the experiment also very hard to see in the variants, but in the computational we are able to see a more stable version of these like variants. And another interesting thing, because interaction that we are able to do, we are seeing that the reason, because like the responding or responder non-responders are mostly related to interaction differences, and we are able to see that the tumors are interacting with CD8 T cells that corresponding mostly to a responders. But in non-responders, instead of like CD8, they are mostly related to CD4, which are more related to regulatory T cells or like immunosuppressive T cells instead. So this probably gave us some answers about like why some of the patients are not able to respond to this immunotherapy. Okay, so here I try to convince you that we have a model that can do a lot of like tasks, and we also confirm from different scenarios, not from the synthetic data. We also showing some other like real cases across this like transcriptomics, proteomics, metabolomics, and epigenomics as well. We also having the data sets across mouse to human, and across Alzheimer's to the brain tissues, hippocampus, as well as the tumor microenvironment. Right. So with that, I want to thank my collaborator in these projects, especially from the Enable Medicines, Alex and Matthew, as well as like Ben and Michael and Joseph. I also want to thank one of my close collaborator from ARC Institute, Jintian, who gave us like a lot of insights on the epigenomics analysis, and my advisor as well, Steve, as well as like Elham. I think this project is also initially like inspired by Starfish, and yeah, and also really appreciate these opportunities to present my work and have discussion with people here. Thanks for Justin and Nathan for organizing it.
Thanks so much. That was
Thanks,
an
you.
awesome talk.
Thank you.
I was super ambitious as usual, and I think you accomplished a lot of things through the model. I was wondering if we could, I guess, okay, we're fielding some questions from the chat. Maybe if you could go back to like the graphical model slide, I think that would be the best to serve as like a reference.
Yeah, sure.
So one question from Mark earlier was depending on what the attention layer um, is serving as a, okay, so I guess the question is kind of like, um, how does the graph attention network fit in? Like, what is attending to what? And what is the reason that's an intuitive explanation for how cells are interacting with each other?
Yeah, so because I think we want to utilize the information that like not just like the cells individually, like we want to know what the neighbors are actually affecting it. So when we getting this like the spatial variable here, we also want to get some other information from their neighbors, and then the graph attention networks can give us because we also have the edges into different like nodes and that is represented cells. So we have actually some information that's from other nodes. So the graph attention networks are able to help in the inferences part for inferring the spatial variable here, mostly related to related to ZI.
So what are the nodes exactly in the graph? Is the node
Oh,
at the cell level or at the spot
yeah.
level?
Sorry, I think I need to discuss in detail actually. I don't know if this one or this one is better. Basically,
Mm,
yeah. So yeah, individual is like cells. So because the whole model is like actually focusing on the cells instead of like spots.
mm,
But we know, for the spots, like we having a in parallel, like we assume there have some cell as well. And so for individual eye, the cell eye, we having a graph, like GI, and then for I, we having their protein, for example, or like modality 1, and we have modality 2. That is like assumed actually because they're not observed, but we have observed version of like aggregated one. So in in the uh in the in the generative or in the inferences part, we as like sum up all of these inferred version together. Yeah. Yeah.
okay, I see. So mainly the edges are between cells, not
Yeah.
the like spot to the cells.
Yeah. Yes,
Got it.
it's the edges between the cells and rely on the cell version modality.
Okay, I see. And we have questions from Eno. I feel like you kind of answered the second question about the
Mhm,
tension.
mhm.
Um, and the first one is about like, okay, question's a little complicated actually. But maybe you can read it and interpret it. Otherwise, maybe you can um, unmute yourself
Yeah,
and clarify anything.
yeah. Maybe can you just like
Oh, yeah, thank you.
mention? Mhm,
I'm just wondering, like, because VAs usually don't have explicit constraints on the latent manifold. So I was wondering
mhm.
for the design of 1D trajectory, is it possible that like different trajectory can reconstruct the same observation?
Hmm.
Say, run the
Identifiability.
model twice, is it always go
Mhm.
from cortex to the two end trajectories? Thank you.
Yeah, that's a very good question actually. I run it multiple times, and then I mean I always get like these directions, not from cortex to medulla, but I got sometimes like from medulla to cortex. Yeah, but
Hmm.
overall, like it was showing the tendency like from one significant to another significant. So I would say, I mean, we don't know what exactly the infer we are, but like we have to figure it out like after we got this V, and then we will need to know what this V means. And I guess like in different data set, they will mean different as well. And I would think maybe in the Q-merge cases, it's probably like more difficult to identify. Yeah, but in our data in our model we actually in our data set in the last cases we actually find something interesting, yeah, but I don't present because I think it's too detailed. But I agree that this is not always fixed values and it will change and the direction will change too. Yeah.
Hmm. Um, Last question from me. I was wondering with the loss, there's a lot of different components of it, and I think I saw you have coefficients for each component of the loss. I was wondering how do you go about doing model selection or like hyperparameter tuning? Because
Mhm.
in addition to having a lot of loss coefficients, you also have a lot of tasks that you're trying to accomplish. So I was wondering what exactly was the metric they used to determine what was the best
Yeah.
version of the model.
Yeah, yeah, I agree. So I do kind of like met a lot of like difficulties when trying to finding the best parameters like for the model. But right now I would say we have fixed it and not really need to change the hyperparameter.
Mm.
The only thing for that to change is I think is related to how the sizes of your spot and cells look like. Because as in some of the cases, for example, in the data we have here, um, the results is not like perfect yet because the resolution differences like in the space is very different. Like here is, I guess,
Yeah.
more than 10 or probably, which is not ideal. And we're making the model maybe not like able to finding a better way. They mostly like collapse into the reasons. So yeah, I think it has limitations in that, and I try to have some like adjustments and just not like manually, but try to figure out something. But right now I have just like one condition that consider the ratios between the size of the spots and the cells, and we'll adjust a bit on here because, for example, the contrastive loss is trying to try to making that more heterogeneous, and then the graph version is like making the more smooth. So there's like a
Yeah.
balance between the model learning. Yeah,
I
it
see.
really depends probably what exactly you want from the data. So we can try to adjust a bit between these two. But yeah, for now we are able to find some like balance.
I see. Any other questions?
Oh, I have one. Sorry, maybe it's last minute, but just one quick question. Could you point me to the term? Because I see there are like four, or in your preprint, there are four loss functions or loss terms in the loss function, the learning optimization, that sector. Could you point me to the term that kind of take care of that the graph-based attention network? Because I cannot find
Oh,
way to that one.
I okay. So I think it's in the masters. I mean I can maybe. Would you mind to send me email and I will maybe point out that in the
Yeah.
exactly text. But I think we have it in the masters.
Okay. Yeah.
Yeah,
Yes.
it's probably in the recon. I think in the last part. Yeah.
Right. I haven't found it yet, but yeah, maybe if you could point me to that place, that would be much better.
Mhm. Yeah,
Would
of course.
you share my email with you in the like a DM or something, or chat?
Yeah, I can send it in a chat.
Okay. Cool.
Okay. Thank you.
Nice. That's nice. Thank you so much.
Yeah, of course.
All right. If there's nothing else, then I think we can wrap it up here. Thanks so much again for a great talk, and it was lovely to hear all the details about what you accomplished with Coral.
Thank you so much, Justin, and very great discussion with people, and I'm very excited. Thank you so much, everyone, for coming.